This is lecture 4. In lecture 3, we talked about the structure of skeletal muscle and looked at a lot of the proteins inside of a sarcomere. In this lecture, we'll look at how the sarcomere is shortened, so how a muscle contracts. That's cross-bridge cycling or sliding filament theory. And then we'll look at some basic factors. I mean, just the various basicest factors that contribute to force production. And we'll go over how the length tension relationship works. But let's start with the structure of a myofibril. Remember, a myofibril is a string of sarcomeres. We talked about this in lecture 3, but there's a bit more to chat about. Under a microscope, you'll see what looks like Beetlejuice sleeves, these alternating light and dark bands. The A band is your myosin. It's all of it. It's all of your myosin. And some actin is in there too. But that's where all of your myosin is. It's dark under a microscope. The A band is dark. The I band contains no myosin at all. It's light under a microscope. So you get these alternating dark and light bands, dark where there's myosin, light where there isn't. And then the H zone, this is part of the A band. It's just the part where no actin at all lives. During contraction, it's the I bands that get squished, not the A band. Um, Except that in the H zone, that like basically disappears because you shove a bunch of actin into it. But while a muscle is contracting, the A bands stay constant while the I bands get squished. And those squishing I bands, that's what brings the Z lines or the Z discs closer together. This is just more of the same. The A band is your myosin end to end, the full length of your myosin. That's your A band. The H zone is the part of the A band where no actin is. And the I band is the part of sarcomeres where no myosin is. The M line is in the exact center of the sarcomere, and the Z discs or Z lines are in the middle of an I band because I bands, these stretch across two conjoined sarcomeres. One I band is part of two sarcomeres. It does get a little bit more complicated with the various proteins involved, but you don't need to know this much. All you need to know is the Z disc, the M line, the I and A bands, and the H zone, and actin-myosin, troponin and tropomyosin, titin and nebulin. And it doesn't look like alpha actinin is shown here, but know that protein too, alpha actinin. So let's start cross-bridge cycling or sliding filament theory very simply, right? We have a row of sarcomeres. They might be 2.2, maybe 2.4-ish micrometers in length each, and we're going to contract them. We're going to contract the I bands of all of them. Each sarcomere's I bands, these get squished together a tiny bit, but across the whole muscle, it ends up being a lot of movement. Just for the sake of reference, you don't need to know this. What the M and Z stand for, they're both German. Mittelscheibe. It looks like a guy drinking beer there, but it's actually a feminine noun. It means middle disc. Mittel, Scheibe, Scheibe is disc. And then Zwischenscheibe means between discs. And nothing you need to know, but if you're wondering what these letters stand for, H zone, H is also German for heller zone, which means brighter. And then the A and the I and A are isotropic and anisotropic. And again, you don't need to know those things. What you do need to know is that there are two Z discs or Z bands, Zwischenscheibes, and these are the outer borders of a sarcomere. And in the middle of a sarcomere, in the Mittel of the Scheibe, there's an M line, and myosin pulls on the actin to draw the two Z bands together toward the midline. The badly drawn humanoid there is myosin pulling on a couple of actins. But let's talk about how it pulls. Sliding filament theory refers to the filaments actin and myosin. These are your contractile proteins. Myosin heads, the head of the myosin heavy chain binds to actin. This bond is called a cross bridge, so that's where the name cross bridge cycling comes from. And once bound, myosin ratchets along actin. It bends its neck, tugging the actin toward the M line, toward the middle of the sarcomere. So remember tropomyosin from the last lecture, a long protein that winds around actin. Think of it like a vine coiling around the actin. And along that vine are berries, occasional berries growing along the winding vine of tropomyosin. Those berries are troponin. And troponin is a complex of proteins. There's troponin C, I, and T. We'll talk about those in just a minute. But first, tropomyosin, that long vine, is capping all of the myosin binding sites along actin. Myosin doesn't have access to those sites, not until tropomyosin moves and those sites are exposed. And to do that, to cause tropomyosin to move, calcium needs to bind to troponin. So here's the troponin complex. There are three subunits, C, I, and T. Each of them is responsible for a specific function of troponin. Troponin T is what binds troponin, the complex, to tropomyosin. It's like the stem of the berry. T is for tropomyosin. Troponin T is bound to tropomyosin. Troponin I is what binds the troponin complex to actin. I is for inhibition. Troponin I holds a tropomyosin in its place, covering the binding sites that myosin wants access to. And then there's the troponin C, which is what calcium binds to. C for calcium, troponin C. And this is bound to troponin I. And when the calcium binds to it, when the calcium binds to troponin C, it changes its shape. There's a conformational change and a dislocation of troponin I. So tropomyosin then moves, and that movement exposes the binding sites on actin, which is what myosin will bind to. In the upcoming lectures, we'll talk about how calcium gets released, why it's suddenly binding to troponin C. But for now, just know that it's stored in a sarcoplasmic reticulum or the sarcoplasmic reticula, that's the plural. This is where the muscle stores calcium, and contraction of the muscle is dependent on the release of that calcium from the sarcoplasmic reticula. Once the calcium is released, it binds to troponin C. That causes the shape change, right, in troponin, which results in the tropomyosin shifting, which exposes the myosin binding sites on actin, and those myosin heads are then free to bind to the actin. But this requires energy. It's not free in that sense. So to perform cross-bridge cycling, you have to consume energy in the form of ATP, adenosine triphosphate. That's adenosine, phosphate, 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 right? Three of them. You chop off one of the phosphates to make ADP, adenosine diphosphate, adenosine phosphate, phosphate, right? Two of them. And on the head of a myosin, there's an ATP ACE, ASE. ACE, this is an enzyme. Whenever you see ACE, think enzyme. And this particular enzyme breaks down ATP. It chops off the phosphate, one of them. That's called ATP hydrolysis. Hydrolysis. You're lysing ATP with hydro, which means you're cutting it up with water. And water is H2O, right? There are two hydrogens and an oxygen in water. The oxygen in one of the hydrogens, one of the two hydrogens, binds to the phosphate that you just chopped off. You cap the POOO with an OH, and then the other H from water, H2O, there's two of them. The other H floats off as a free, as a hydrogen ion. And that's the H of HLTH, meaning that's acid, and we'll talk about that later. But this is what provides the energy to do cross-bridge cycling. But that initial bond between actin and myosin is weak as long as ATP or ADP and the newly disconnected phosphate are attached to the myosin head, the bond is weak. The bond between actin and myosin, right? That's weak if ATP is still bound, or if ADP and the phosphate are bound, you will have a weak cross bridge. And to do cross-bridge cycling, to slide those filaments in sliding filament theory, you need to transition to a strong bond. The bond between actin and myosin needs to be like a real estate agent's handshake or like a father meeting his daughter's boyfriend for the first time handshake. To do that, the phosphate is released. PI means phosphate, inorganic phosphate, inorganic because there's no carbon in it. By releasing that phosphate, the capital P, lowercase I, by releasing that, you transition to a strong bond. As long as just ADP is bound to the myosin head or no phosphate, um, just like, nothing at all, you have a strong bond. So ADP without the phosphate or nothing bound at all, you're going to have a strong bond between actin and myosin. So once the myosin head releases the phosphate, it transitions to a strong bond. It cranks. It completes its power stroke, yanking along actin, bending its neck. But it doesn't move very far, just a tiny bit with each power stroke. Then it needs to let go of the binding site it's on and reach for another, crank along that one, release again, and reach for another, crank along that one, release again, reach for another. If the bond is strong between actin and myosin, it can't release. So you have to bind another ATP to the myosin head to transition back to a weak bond, then it can release. Hydrolyze the ATP and just repeat the process over and over and over, cross bridge cycling. And the muscle it'll continue to contract. It'll continue to do this until calcium is put away and the tropomyosin slides back over the binding sites along actin, blocking myosin's access. Then there are other things that can mess up cross-bridge cycling too, like if the ratio of ADP to ATP gets too high and you have too many phosphates lingering, this can impair the function of actin and myosin. So, calcium turns on the lights, but other factors can function as dimmers, and we'll talk about those factors in detail at the end of the semester. But this is a step-by-step look at cross-bridge cycling. These aren't formal steps. I just wrote it out and assigned arbitrary numbers. So don't memorize how the steps are numbered and categorized. Just know the sequence of events in sliding filament theory. Moving on from muscle contraction and into a new but related concept, we have the length tension relationship in skeletal muscle. What the length tension relationship is showing is that a muscle produces both active tension, that's cross-bridge cycling, and passive tension. Um, for passive tension, all you need to know is that tighten provides most of that stiffness and a stretch. But there's also contribution from the mesomes, right? Epiperi-endomysiomes, and other structural proteins in the muscle. Active tension is what we're going to focus on for the length tension relationship, though. And active tension is generated by those actin myosin bonds by cross bridge cycling. And here with active tension, think about how much force you can generate at different points in the range of motion. So take like a bicep curl. If your arm is fully extended, your actin and myosin will look like how it's pictured in the far right of the diagram. The myosin heads won't be able to reach the binding sites on actin. The sarcomeres will be too stretched out, So the amount of cross bridges you can form will be reduced. So contraction will be weak. A skeletal muscle isn't very strong when it's all stretched out. Contract it a bit though, and you'll be able to produce more force because there's more overlap between the actin and myosin. More myosin heads can reach the binding sites on actin. In the middle of the range of motion, you tend to be able to produce the most force. But if you keep contracting, keep flexing that bicep until your hand, meets your shoulder or nearly meets your shoulder, then it'll be weak in that position too. You'll have basically eliminated the H zone. And again, there will be a reduction in the number of total actin myosin bonds that can be formed. When the sarcomere is in like a neutral, right in the center, the neutral position, in the middle of your range of motion, that's when you can produce the most active force, because that's where you can generate the most cross bridges. Now, remember from the last lecture when we talked about sarcomerogenesis and sarcomerolysis, the insertion and deletion of sarcomeres. If you're constantly training in an extended position, the right side of the diagram, it would help your sarcomeres be in a more neutral position if there are more of them, more sarcomeres in series to help buffer that stretch. So sarcomerogenesis occurs as an example of specificity of adaptation, and vice versa. If you're constantly training in hypercontracted states, the left side of the diagram, then sarcomerolysis would help your sarcomeres be in a more neutral position when you're training through that range of motion. So again, this is just specificity of adaptation. In this case, it's angular specificity, which is a concept we'll discuss more later. So like where, I mean, very broadly, very generally, do we get our strength from? First, it's the total number of muscle fibers being called upon. That matters. Do you have 1 million muscle fibers? Every fiber in your medial gastroc, are you recruiting all of those? Are there 500,000 fibers contracting? You're still in the medial gastroc, but only half the fibers are participating. Are there only 100,000 fibers contracting, 10% of the fibers in that muscle? The force production will vary according to the number of muscle fibers called upon. Second, muscle fibers, they're not uniform in size. Not all muscle fibers are the same. So some have a ton of actin and myosin in them, lots more myofibrils, and others have fewer. Other fibers are smaller and weaker. So fibers themselves determine force production. And then third is the function of the cross bridge uh, cycling of the cross bridges themselves. I mentioned earlier that it's not just the presence of calcium that matters. That turns on the light, but other factors can serve as dimmers. And calcium release and reuptake can be affected too. So there are a lot of physiological explanations why a particular fiber, regardless of its size, might not be contracting effectively. And then there are other contributors to force production too. This isn't a complete list, but we're going to get into those in future lectures. Coming up in the next lecture, we'll start looking at the nervous system's control over skeletal muscle. But for this one, for this lecture, here's what you should know. It's not just definitions of the vocabulary, but how things work. Make sure you can understand how these things work too. And then lastly, you should be able to label diagrams. This tends to be a little bit easier than understanding functions and processes, but be able to label diagrams as well. And that's it for this lecture. I'll see you in lecture 4, where we will start this is lecture 4. I'll see you in lecture 5, where we'll start a discussion of the nervous system.